and welcome. I'm trying to learn cytology today, exocytosis, and after we've done it, we're gonna do extensocarpi nerve strength training. And if you participate in the extensocarpi nerve strength training, then it's really wrong. Exocytosis, as depicted in figure 313. Wait one. Through 314. Exocytosis involves the movement of materials out of the cell in vesicles that fuse with the plasma membrane. The ejected material, who's creating these cells? These clefrin, and who's creating... Okay, we said... Yes, but who's creating the phospholipids of the plasma membrane and the glycolipids? Because it can't... Yes, it can do cells. Where are we coming from? How does this cell is being... Okay, that makes sense. Okay. The ejected material may be either a waste product or a useful secretory product. All cells carry out exocytosis, but it is especially important in two types of cells. Nerve cells, which release substances called neurotransmitters, and secretory cells, which secrete digestive enzymes or hormones. Okay, there are some who bring them through vesicles out of a cell, exocytosis, things out. And when it's not so accidentally, these are, for example, neurotransmitter. They are bringing them things out. We did with extreme neurology. And we saw, I think it was acetylcholine was the one. It comes out. It goes over this synaptic vesicle over the little gap of the synapse, and then to another cell, the target cell, where it opens there something, and then the electro signals can go through and create there as an impulse a stimulus which will then contract the muscle, and that way the nerve signal has been transported and the muscle is moving. It was something along the line. And also secretory cells releasing enzymes and hormones. During exocytosis in secretory cells, membrane enclosed vesicle called during exocytosis in secretory cells. So are not all cells doing that? So if we're talking about cells doing that, then they were talking about the phagocytosis, and then suddenly they meant these were the white blood cells doing. It seems like most blood cells aren't doing it. And now are that only the secretory cells doing, and most cells not? During exocytosis in secretory cells, membrane, I'm basically asking, what will you explain? Are they explaining that for cells, or explaining that just mechanism which some cells have, or cells have? Some cells of them. And there is not a single cell which encompasses all of them. During exocytosis in secretory cells membrane, so if you have now, let's say we would recapitulate, is the right word, all what was written here now, and say a cell has this, it would make absolutely no sense. It would be like an animal consisting out of 10 different animals at the same time. An elephant, a giraffe, and a donkey, a dog, and a cat together in one animal. During exocytosis in secretory cells, membrane enclosed vesicle. I don't understand the sentence, but problem. During exocytosis, so something comes out. In secretory cells, cells which are bringing something out. These membrane enclosed vesicles. It's a vesicle around which a membrane is. Okay, that's the thing we're talking about all the time about. That is vesicle, and around is the membrane. Membrane enclosed vesicle called secretory vesicles. Okay, why not? Form inside the cell, fuse with the plasma membrane, and release the contents into the extracellular fluid. Okay, these secretory vesicles are forming. Did we say it here? No, we didn't say it here, but it could be something like that. There it becomes when, maybe actually from the late industry, but maybe not. Maybe that is another type of cell. So it can become when it forms when the stuff inside of a cell, it forms this. Yes, good question. How does the cell get the product out of it? How is it delivering it? That is the answer. No, we have the answer. The answer is when it goes in around, makes again a cell, a vesicle, covered again with the membrane, so that it can come again to the plasma membrane and sort it back in and out there again. Portions of a plasma membrane that are lost through endocytosis are recovered or recycled by exocytosis. We said it here for a second time. Is it necessary? How difficult is it for the cell to make new ones? Who is making these new ones? Is it helpful? Is it necessary or just accidentally? Or whatever. The balance between endocytosis and exocytosis keeps the surface area of a plasma membrane relatively constant. The surface area. So the first thing, maybe it goes also very fast. They were talking in the previous part about some channels, and then suddenly they said through a single of these channel pores can go 1 million, I think it was potassium, per second through. So when we're talking about there is a channel and there can come something through, it's not like for ships, maybe 1 every 5 minutes or 1 every half an hour, 1 million per second. So if we're talking here about that, this vesicular transport, is it something maybe occasionally happening? Or is it something like that happens once per second, tens, 1,000, 1,000,000 times per second? Then it would make a difference when it is sorted back to the other side if the cell isn't shrinking or something. What happens if it happens? Can the cell then die? Is the cell then forming cancer? What is the beginning of cancer? The balance between endocytosis and exocytosis keeps the surface area of the plasma membrane relatively constant. Membrane exchange is quite extensive in certain cells. Okay, exchange. So we're saying some cells are doing that a lot, but from once, how did if it is a secretory cell, how did it get now all the plasma membrane into the center of it, so that it can form a secretory vesicle and bring it to the outside? Or do all cells have some place where it goes in and out? It makes sense with the last sentence where we said it is balanced. So the secretory cell need to have a lot of import so that they can make inside of the cell these secretory products. And there are less vesicles going out if we got less import, and if we get less import, we don't have so much, but we don't also need so much. Okay, secretory cells of the pancreas, for example, recycle an amount of plasma membrane equal to the cell's entire surface area every 90 minutes. Okay, it's not like 1 million or something probably, but I don't know. Who knows how many there were? Maybe there are quadrillion of cells of whatever they are on the outside, these chlorine-coated pits, but they're suddenly not talking about anymore about them. Is this still about chlorine-coated pits? Here are no chlorine-coated pits on these pages, just a minute. Yeah. No, every 90 minutes. Table 3.1 summarizes the processes by which material is transported into and out of cell. 3.1, where's 3.1? That is 3.1. Osmosis, diffusion through the lipid bilayer, diffusion through membrane channels, facilitated diffusion, active transport, primary active transport, secondary active transport, vesicular transport, endocytosis, receptor-mediated endocytosis, phagocytosis, pinocytosis, and exocytosis. That was the... Why did we make it on the next? Okay, maybe there was some space. If you were here for cytology, you can go directly to the next part of cytology. Let me start to go back to the beginning of the chapter with the transport across the plasma membrane, and then this go through the whole thing, what we had there also listed, and try to make a presentation about each of these little things there. If you stay here and participate in the extensor carpal strength training, then it's very honest. Good as still stand, arms up, hands onto fists, bring them down. And up. We're having 3 routes. The first one is that we're stopping immediately as soon as there's pain. Hands are good and down. The second one is that we're going only as far as we can go with a little bit of it. And the third one is that we're not overdoing it, we just want to strengthen, not to exhaust. And now you can use a sock, a t-shirt, a towel, or a towel, press onto it, and down. Press equally with all 5 fingers. And up again. And down. And change. Press. Down. And again. And now just press and wheel time. And again. And change. Press in. And out. And press on top. And again down. Change. Press up. And down. And just press in neutral position. And also on the other side. And that was the extensor couple now strength training. I thank you very much and